Actinomycetes. These are the organism that has the structural resemblance with the fungus as well as the bacteria. That's why they are called as the true bacteria. And the organisms that are involved in the actinomycosis of the cattle and the human. So these organisms are the traditional forms and shows the transitional forms between the fungus and the bacteria. The resemblance to the fungus is that they produce mycelia like filaments, the branching filaments, and the characteristics of the bacteria is that they are thin and they show a cell wall having a muramic acid and a prokaryotic nuclei as well as they are sensitive towards the antibiotics. Now because they have the characteristic feature of the fungus as well as the bacteria, they are called as Actinomycetes, and they are true bacteria resembling the fungal cells. And here these organisms show relation with the mycobacteria as well as the corynebacteria. They are gram-positive. Some of the species are acid-fast in nature. They are non-motile, non-sporing, non-capsulated, can break down into branching filaments or into the coccoid elements. They are mostly free-living in the soil, and the organisms which are of medical interest belonging to the Actinomycetes are usually the anaerobic Actinomycetes, Rothia, Nocardia, aerobic Nocardia, Streptomycetes, etc. And these organisms that are mostly involved in the human pathogen are anaerobic Actinomycetes. So Actinomycetes which are anaerobic, non-acid fast, are the one that causes the infection in the humans, as well as the other species belonging to this Actinomycetes are the Nocardia, which are aerobic in nature and they are acid fast. And apart from these two organisms, the other causative agent of the human disease is the Streptomycetes, but it has more importance towards the production of its antibiotics rather than the causation of the disease. It is given importance because it produces most of the antibiotics. So the human infections are caused by two important group of Actinomycetes, which are anaerobic Actinomycetes and the aerobic Nocardia. Coming to the Actinomyces, this was first identified by Bollinger in 1877 in a lumpy jaw, which was seen in the cattle infection. So it was called as actinomycosis. So it was in this lumpy jaw, this organism was first described by Bollinger. The term Actinomyces was given by the scientist Haas, who coined it as the ray-like appearance. Sun ray-like appearance was seen in the granular lesions that are produced by this organism. So because of the ray-like appearance they show, the term Actinomyces was given, that is ray fungus. Now the organism was first identified or isolated from the human infection was by Wolf and Israel, who were successfully able to isolate the organism from the human infection and transmit it to the experimental animals such as the rabbit, guinea pigs, etc., which showed the same type of the disease that is caused in the experimental animals. So it was first isolated from the human infection by Wolf and Israel, and hence the organism that was isolated was given name as Actinomyces israeli, which causes infection in the humans. The organism which is involved in the causation of the disease of the cattle is Actinomyces bovis. Coming to the disease, actinomycosis, it is a chronic granulomatous disease which the characteristic feature of this is it causes indurated swelling, especially involving the connective tissues. Suppuration is seen along with the presence of sulfur granules. The presence of sulfur granule is a characteristic feature of this particular disease and helps in identification of the organism in the lab diagnosis. It is usually caused to the skin, which can lead to multiple sinuses. So there is a discharging sinuses which contains the granule that helps in identification of the organism. It is mostly a human disease and it is endogenous in nature because these actinomyces are usually the commensals of the humans. They are present in the mouth, intestines as well as the vagina. So any damage to this area can lead to the infection with this type of organism. That is, if there is any foreign body damage or poor oral hygiene, then this organism is able to invade that particular tissue and cause the infection, leading to actinomycosis, and hence it is called as an endogenous infection. And the tissue invasion is only when there is damage to the these areas that helps in the invasion of the organism and cause the disease. So the most prominent organism that is involved in actinomycosis is Actinomyces israeli. Apart from that, the other species are also involved, called as Nestlandi, Actinomyces nestlandi, viscosus, and Actinomycetes odontolyticum, Myri, Gerexoni, and Propionibacterium propionicum. This particular disease is said to be a cooperative disease because it is not caused by a single type of organism but in combination with other bacterial diseases, mostly caused by Bifidobacterium, Actinobacillus, Actinomycetum comitans, Ignella, and Ignella corrodens, and we have Haemophilus and bacteroids. Apart from that, fusobacteria and staphylococcus and anaerobic streptococci can also help in causation of the disease, because this organism does not cause disease alone, but with the help of other bacteria it is called as a cooperative disease. Now coming to the clinical forms of this disease, there are 4 clinical forms of this disease that have been identified affecting different parts of the body. First is a cervicofacial, which involves the cheek and the submaxillary region. Next is a thoracic, which involves the lung, where the pericardium muscles can also be involved, or the pleura of the lungs are usually involved, where the inflammation can take place because of this particular lesions, and can progress towards the chest wall. And coming to the abdominal, the cecum is mostly involved along with the neighboring tissues and the abdominal wall. This infection can also be transmitted to the lungs via the portal vein. Next is the pelvic actinomycosis, the pelvic form, which is seen only in the female who uses intrauterine devices. So it is because of the intrauterine devices, which is not sterile in nature, can lead to the infection of the actinomycetes. Next is this organism is not only involved in this 4 clinical form, but is also said to cause gingivitis and periodontitis, that is the gum inflammation when there is a poor oral hygiene, involving the sublingual plaques and leading to root surface caries. The presence of mycetoma can also be seen in this particular infections. Coming to the lab diagnosis, the lab diagnosis is usually done by the granules that are formed during the lesions in the particular disease, and appearance of the granules or identification of the organism in the microscopy. Next is isolation of the organism in the cultural media. The specimen that is mostly used is a pus in this particular disease, and in the pulmonary diseases the sputum can be taken as a specimen. And the observation of the sulfur granules in the microscopy helps in demonstration of the organism and identification of the organism. The sulfur granules can be collected from the discharging sinuses, and they are usually taken in a test tube mixed with the saline, and when the granules settles, they can be extracted with the help of a capillary pipette, or directly we can collect the granules from the discharging sinuses with the help of a sterile gauze pad. Now these granules are collected and taken on a cover slip, and then they are crushed between the slide and the cover slip and observed under the microscope. The appearance of gram-positive thin filaments or thin gram-positive filaments surrounded by the club-shaped structures which gives a sun ray-like appearance, and hence this particular characteristic feature is taken as a positive for the actinomycosis. That is Coming to the lab diagnosis, how do we identify this organism in the lab? We can go with the demonstration of the organism, that is from the sulfur granules that they produce under the microscopy, or isolation of the organism in a cultural media or a specific media that is used to isolate the organism. Coming to the specimen that is used over here is a pus, because there is a multiple discharging sinuses that are related to the disease. The pus can be taken as a specimen, and in the case of pulmonary disease, the specimen is a sputum. Then demonstration of the sulfur granules can be done in the laboratory. The sulfur granules are mixed with the saline in a test tube, and the precipitate that is usually formed at the bottom is taken with the help of a capillary pipette and observed under the microscope. Apart from that, direct sulfur granules or the granules can be collected from the discharging sinuses with the help of a sterile gauze pad. Once collected, they are usually placed on a cover slip and crushed lodged between the slide and the cover slip and observed under the microscope. Here, the characteristic feature of this is under the microscope they appear as thin gram-positive filaments that are surrounded by the club-shaped, radiating club-shaped, and hence giving the structure similar to that of the sun ray-like appearance. And the clubs that are seen under the microscopes are thought to be due to the antigen-antibody complex. So irrespective of whatever it is, the sun ray-like appearance under the microscopes helps in identifying the particular organism. Next, coming to the culture, this organism can be very well grown on liquid as well as a solid media. The media that is used to grow this organism is a thioglycolate media. Now, because this organism is anaerobic in nature, they have to be kept at anaerobic condition at 37 degrees centigrade. So, two media that are used is a thioglycolate medium, which is liquid medium, and brain heart infusion agar, that is a solid medium. So, in a liquid medium, that is a thioglycolate medium, the sample is inoculated, that is a sulfur granule or the pus has been inoculated directly and incubated at 37 degrees centigrade in anaerobic condition. The Actinomycetes bovis usually forms turbidity in liquid media, whereas Actinomycetes israeli usually forms fluffy balls at the bottom. So, the presence of fluffy balls at the bottom indicates a positive reaction. On a solid media, that is a brain heart infusion agar, the organism Actinomycetes israeli usually forms spidery colony within 48 to 72 hours, which on further incubation for 10 days can result in heaping of the colonies, showing white, irregular or smooth colonies which are large in nature. Now these colonies are usually further ahead they are confirmed with the help of microscopy and biochemical reactions. Apart from that, we can use more sophisticated techniques such as fluorescent antibody techniques. Gel diffusion and immunofluorescence is mostly used to distinguish between Actinomycetes israelis and the other filamentous organism that produces similar type of granules in their disease. So in order to distinguish that, we can go with the gel diffusion and immunofluorescence. Now coming to the epidemiology of this disease was thought to be a worldwide disease, but the cases have completely come down after the treatment with the antibiotics, and it is now only disease of the rural areas affecting the agricultural workers, etc. And among them, young males are mostly affected between 10 to 30 years. The reason for this is unknown, but it is said that only the young people with a poor oral hygiene are said to be infected with this particular disease, and 60% of the diseases is because of the cervicofacial and 20% is abdominal. And in women it is usually causing pelvic actinomycosis because of the use of intrauterine devices. Coming to the treatment aspect, penicillin and tetracycline for a prolonged period of time can treat this particular disease, and if not treated, then can go with the surgery to eliminate the infection. Coming to the other group of organisms, that is the Actinomycetes, the aerobic form is a nocardia. Here the organism nocardia is aerobic, gram-positive, but mostly it is acid-fast in nature. So among the nocardia, the group of organisms that are acid-fast are nocardia asteroids and brasilensis. These nocardia are usually present in the soil, and they causes the exogenous infection rather than the Actinomycetes, which causes the endogenous infection. This organism is the one that causes exogenous infection. And the three important types of infection caused by this organism is cutaneous, subcutaneous and systemic. And the organism involved in the causation of the disease are N. asteroids, N. brasilensis and KBA. Coming to the cutaneous infection, this is characterized by local abscesses, cellulitis, lymphocutaneous lesions. These are the three important characteristic features of the cutaneous infection. Now coming to the subcutaneous, it is also called as Actinomycetes mycetoma or actinomycotic mycetoma, which we will be studying in the next slide in detail. Now coming to the systemic infection, which is caused by N. asteroids, which is mostly involved in the systemic nocardiasis, involving the lung lesions, or it also causes the pneumonia and the lesions of the other important organs. Metastatic involvement or the metastatic manifestation can also be seen in the brain, kidney and the other organs. Now coming to the lab diagnosis of this particular infection, that is a nocardia, microscopic observation from the lesions containing branching filaments of the organism can be taken as a prognostic test or the presumptive test, which can be further confirmed with the help of cultural media. This organism grows very well in an ordinary media, and the type of colonies that they produce on an ordinary media are dry, granular, wrinkled colonies, and are able to produce pigment that ranges from red to yellow. So the presence of pigmented colonies helps in identifying this nocardia species, and the treatment is usually done with the help of quatrimoxazole, which is given for a prolonged period of time. Apart from that, minocycline, amic acid and cefotaxime are other choice of antibiotics that are used to eliminate the infection. Now coming to the actinomycotic mycetoma, that is a subcutaneous infection of this nocardia. It is a localized chronic granuloma that is formed due to the production of the lesion in the subcutaneous layer or the deep tissues of involving mostly the foot rather than the hands. So very rarely we can see on the hand, but mostly they cause the deep tissue inflammations of the foot. Apart from that, they also present tumors and multiple discharging sinuses. This clinical syndrome of this particular disease was first seen in Madura by the Gill, and hence the organism was called as the clinical syndrome was called as Madura mycosis. And the mycetoma are the ones that are usually produced by the fungi, but apart from fungus, there are certain bacteria that are also able to produce the mycetoma. For example, the anaerobic staph that has the ability to produce the mycetoma-like structure called as botryomycosis during its infection. The lesion is called as a botryomycosis, and as told, mycetoma is only a characteristic feature of the fungus, but also caused by the bacteria. The bacterial involvement over here is from the Actinomyces. The species involved over here is Actinomyces israeli and Actinomyces bovis. And Nocardia asteroides, Nocardia brasilensis and Nocardia caviae. And Actinomadura, that is Actinomadurae and Actinopelleteri. And coming to the Streptomyces, Streptomyces somaliensis is the organism that is involved in the production of the mycetoma. Now, when you are going for the etiological treatment, that is for treatment aspect, we should very well know which type of organism is usually causing this particular disease. That is, the etiological diagnosis of the disease is very important, because this mycetoma is not only caused by the fungi but also by the bacteria, and hence establishing the causative agent is very important for the treatment aspect. So the way to distinguish between the fungal mycetoma and the bacterial mycetoma is the production of the granules, pigmented granules, that makes us to distinguish between the eumycetoma and the actinomycetoma. In the actinomycetoma, they usually forms white or yellow granules which are very thin in nature, that is 1 micrometer. Whereas the eumycotic mycetoma, the granules that are produced are black in nature, measuring about 4 to 5 micrometer. So this is the way we can distinguish between whether it is a bacterial mycetoma or a eumycetoma. And once it is established the presence of the lesion and the granules, when we can go for the treatment purpose. Next is the Actinomycetes hypersensitivity pneumonitis. So this organism also has the ability to cause hypersensitivity reaction. Hypersensitivity is an exaggerated immune response, and the organism that is involved are mostly the thermophilic Actinomycetes. Example, Phaenia and Saccharomycosporia. These organisms, spores, when they are inhaled, they usually causes allergic alveolitis, which can lead to chronic obstructive pulmonary disease, COPDs, also called as a farmer's lung. So Actinomycetes are the organism that causes actinomycosis, and apart from that, they are also involved in the hypersensitivity reaction in allergic individual, leading to farmer's lung.